Greetings dear students. Let us get introduced to the science called plant biotechnology in this video session. In this session, we will be learning the basic concepts of plant biotechnology, an overview of plant tissue culture, as well as some important terms with respect to plant biotechnology and tissue culture. Plant biotechnology is described as the application of recombinant DNA techniques to crop improvement or to the production of valuable molecules in plants. So it involves a set of molecular techniques that can be used to adapt plants for specific needs as well as opportunities. Most applications of modern plant biotechnology involve the manipulation of DNA sequences originally isolated from other plant species or microorganisms or even animals. This technology began in the early 1970s when it became possible to purify and amplify DNA sequences from bacteria and subsequently from higher organisms like plants and animals. By the next decade, that is by 1980s, various techniques or methods that can be used to transfer the exogenous DNA or foreign DNA into host organisms, which may be again microbes, plants or animal cells, came into availability. These techniques were broadly of two types: those techniques which involved the usage of a vector for carrying the exogenous DNA into the host cells. The vectors could be either bacteria or viruses, and another set of techniques which involved direct transfer of the exogenous DNA into the host plant cells. The purified DNA sequences can be used in two major ways. The first one is the purified DNA sequences may be inserted in the form of coding regions or other functional regions such as promoters and terminators into a plant to confer new or desirable properties to the plants. These properties may be beneficial in situ to the plant as such, or the products of these exogenous genes may be isolated and purified for further usage. The second major application of purified DNA sequences was usage as genetic markers. So these DNA sequences were used to assist selection of chromosome segments, which in turn were representative of certain phenotypic desirable characteristics among the progeny of sexual crosses. This whole process was followed as a part of molecular plant breeding programs. So there are two major applications of purified DNA fragments. One is usage of these purified DNA fragments, either the coding region, that is the gene as such, or other functional regions like the promoters and terminators. So these functional regions are introduced into a host plant, because of which the host plant starts expressing new proteins, which in turn are responsible for new desirable characteristics. So these characteristics may help improving the quality of the crop as it is, or these new products expressed in the host plants may be isolated, purified and used for further applications. So this was the first application of the DNA sequences purified from any of the sources. Another major application of these DNA sequences was their uses as genetic markers to produce improved crop varieties under molecular plant breeding programs. So all the applications of these molecular techniques to plants can be broadly categorized into two, one being transgenic applications and the other being non-transgenic applications. Transgenic applications are those what we discussed till now, that is the improvement of crops by incorporation of desirable agronomic traits giving rise to transgenic crops. They also can be used for production of products in host plants, that is they allow the use of these plants as natural bioreactors. Another set of applications using these molecular techniques are non-transgenic applications. The first and foremost among these being marker assisted selection. So marker assisted selection is a method of DNA fingerprinting which is very similar to what we do in medical diagnostics or in forensic sciences. So these molecular markers such as microsatellites, RFLPs and RAPDs help in identification of certain chromosome segments in the host plant genome at molecular level. These segments in turn are representative of desirable phenotypic or genotypic characteristic features. Then comes another major application of plant biotechnology, that is tissue culture and mass propagation. Tissue culture is the technique of allowing growth or regeneration of whole plants from isolated cells, tissues or organs of plants under in vitro conditions. The tissue culture based applications of plant biotechnology include organ and tissue culture, micropropagation or clonal propagation, somatic hybridization, embryo rescue and production of doubled haploids. The applications of plant biotechnology are not limited to crop improvement. They extend over a wide range of areas including production of pharmaceutical and industrial products. The use of plant biotechnology for production of pharmaceutically important compounds is termed as molecular farming. In this, a gene coding for an enzyme or any other pharmaceutically or therapeutically important protein is isolated from the source and inserted into a plant host, allowing the expression of that target gene or the protein in the plant host. This enables us to produce the target protein in large quantities in a cost-effective manner. These proteins are then isolated, purified and used for their specific applications. There are hundreds of such compounds being produced, including enkephalin, interleukins, so on. Then pathway engineering is another major application of plant biotechnology. For example, you may be knowing vincristine and vinblastine are compounds of chemotherapeutic importance. These are produced in the root cells of Vinca rosea. They are intermediates of certain metabolic pathways. So by engineering that metabolic pathways in such a manner that these products are not converted into the end product of the pathway, we are making the possibility of accumulation of large quantities of vincristine and vinblastine in the cells. So these compounds are then isolated and purified for their specific purposes. Many secondary metabolites like essential oils, perfume components, then flavoring agents and other substances which are generally produced by plants as secondary metabolites have been produced in large quantities by using suspension cell culture techniques. Many other industrially important or edibly important compounds are also being produced using similar techniques. This area is generally termed as molecular farming. Examples of edible products that have been produced in such manners are oils and oleochemicals. It is possible to reduce the polyunsaturated fatty acid content in edible oils by biotechnological methods. Similar applications have been used to produce useful oleochemicals used in adhesives, paints, cosmetics, etc. Enzymes that are being produced are avidin beta-glucuronidase. Polyhydroxyalkanoates are a biodegradable alternative to chemical plastics. Starches that are useful in food processing and in production of packaging materials have been produced using biotechnological modifications in plants. Similarly, antioxidants like alpha carotenoids, phenols, and steel beans are all being produced. Nanobiotechnology is another major application wherein plant biotechnological techniques are being used to synthesize nanoparticles which have varied range of applications. This is also called as green synthesis of nanoparticles. Then germplasm conservation and production of plant-based antibodies are other major applications of plant biotechnology. Plant biotechnology is also being used to study the genomics and modify the genomes in such a manner that they are useful for mankind. So, the biological nitrogen fixation ability of certain microorganisms has been extended, or the efficiency has been increased in those microorganisms which already are capable of doing nitrogen fixation. So this has been done by transferring genes for nitrogen fixation to those species of bacteria which are not naturally capable of fixing the nitrogen. So these are all the applications of plant biotechnology. Moving on, let us have a look at what is plant tissue culture and what are its applications. Plant tissue culture is described as the technique of growing cells, tissues, or organs of plants in an artificial medium under in vitro aseptic conditions. The whole concept of plant tissue culture is based on the fact that individual cells, especially plant cells, are capable of regenerating into a complete organism by giving rise to different types of other cells. So this ability of plant cells is termed as totipotency. The major applications of plant tissue culture include maintenance and production of pathogen-free plants, then rapid clonal propagation of plants like banana, teak, grape, and many others, production of transgenic plants with desirable characters like insect resistance and disease resistance, large scale production of secondary metabolites. The basic understanding of biological processes or metabolic pathways that happen within the plant tissues, production of haploids for further selection. So, under plant tissue culture, basically a plant tissue or organ like the shoot tip, a part of the leaf, nodal region, or a floral organ like the embryo or the ovule or ovary is collected under aseptic conditions. It is then transferred onto a synthetic nutrient medium under laboratory conditions and allowed for incubation. So when specific or required environmental conditions are provided to these incubated plant tissues, they slowly undergo de-differentiation, giving rise to a callus most of the times. Now this callus again undergoes redifferentiation, giving rise to shoots and roots of that species of the plant. So these shoots are subjected to root induction, which are then transferred to the field conditions for further growth. So this is the basic procedure of plant tissue culture. Let us have a look at the important terminology which are involved in plant tissue culture. Explant. Explant is the initial material collected from the parent plant for initiating a tissue culture process. Totipotency is the ability of a plant cell to regenerate into a whole organism or a complete organism. Cell differentiation is the process in which an embryonic cell or a stem cell or a meristematic cell undergoes differentiation, giving rise to specialized type of cells like the xylem cells or phloem cells. Dedifferentiation is an event in which the differentiated cells or specialized cells like the floral cells or the leaf cells revert back to their embryonic status. So this process is called as dedifferentiation. The process of dedifferentiation generally gives rise to an unorganized mass of cells called as callus. Callus, after a certain amount of growth and a period of time, starts undergoing redifferentiation, wherein the undifferentiated cells or the embryonic like cells undergo differentiation, giving rise to specialized type of cells. So this process of organogenesis or embryogenesis from the callus involves redifferentiation. So all these are the events that take place in almost all types of tissue culture. Tissue culture methods are generally named based on the explant that is being used. So callus culture is in using of an explant to give rise to a callus, and from the callus again induce shoots and roots. In meristem culture, meristem is used as the explant. Organ culture includes using embryos, ovaries, ovules, anthers and other organs of the plant for initiating the tissue culture process. Use of nodal regions is termed as nodal culture. Similarly, we have leaf culture. Suspension cultures are cultures in which individual plant cells are allowed to grow in liquid medium. So these are all the different types of tissue cultures. I would advise you to refer to these websites and books for deeper understanding of these concepts. Thank you.